Well, good afternoon, everybody, and Happy New Year. Here we are on January 13th with our first patient webinar series of the year. For those that don't know me, my name is Donald Miyazaki, and I'm the Executive Director at the ALS Society of BC. Very excited for today's presentation. Once again, thank you to our friends at Tanabe Pharma Canada who have come on board to allow us to put these webinars on, and they've come as a presenting webinar series. As sort of a housekeeping note, all of the webinars, once again, are recorded. We'll quickly edit them following the webinar and post them on our website under the webinars tab, and you're able to watch all of the recordings as they get uploaded. Another thing is that we do have a Q&A box. So if you have any questions, please enter the questions in the question and answer box, and we'll get to them as quickly as possible on behalf of anybody who may have some questions. So on that note, as I mentioned, I'm very excited for today's presentation. We opened the webinar series in the summer on research with Dr. Pfeffer speaking on genetics. And today we circle back to a presentation on research in ALS. And I think it's a very interesting and well sought after topic that we keep seeing when we ask for, sort of topic ideas in these webinar series. So we found an incredible speaker today to speak on cellular defense mechanisms in ALS. And I'm going to do a quick intro, and there's a lot of words that are far beyond my education, so forgive me for some of the pronunciations. But our speaker today obtained her Bachelor's of Science in Physiology and Pharmacology and Master's of Science in Pharmacology from the University of Western Ontario, and later got her PhD in Pharmacology from the University of Alberta. After postdoctoral studies at McGill University, she continued her studies at the Montreal Neurological Institute, where she achieved the rank of professor. She is currently Professor Emerita with post-retirement in a post-retirement academic role. Sorry, I think I missed this, but after her postdoctoral studies at McGill University, she continued her studies at Montreal Neurological Institute, where she achieved the rank of professor. Throughout her career, her laboratory has focused on the study of ALS and other motor neuron disorders. Administratively, she has quite a track record. She has held several positions, including Director of Graduate Programs in Neurological Sciences and Associate Dean of Graduate and Postdoctoral Studies at McGill. She's also served with ALS Canada in many capacities as chair of the Scientific and Medical Advisory Committee, member of the Board of Directors, and member of the Medical Advisory Committee. Finally, she served in the roles of President, Vice President, Treasurer, and member of the Board of the Society of Toxicology of Canada, while also serving as a member and chair of the Biological Chemical Defense Review Committee. That's how important it is. It's very important. It's difficult to pronounce, with the Department of National Defense, which obviously is a very high status. Well, she does still have a presence in Montreal, and we talked about she goes to the dentist in Montreal. That's how she's still attached to Montreal. We're very proud to call her, she calls Victoria home, where she spent a lot of the last summers over the past 25 years. But we're very lucky now that she's semi-retired. We can call her a BC, right, where she's serving on the ALS BC board and recently appointed to ALS BC's Project Hope Committee. That was a mouthful, Heather. Here to speak on cellular defense mechanisms in ALS, please help me welcome Dr. Heather Durham. Heather.
Well, thank you, Donald. You did a great job, and now you can relax. It's a real pleasure to do this for SBC and for people across the country and even the world who may want to tune in. And what I'm going to talk to you today about is the large variability in the clinical manifestation of ALS, so that each person's journey is unique. And I think if you are affected by ALS, you kind of know that already. But it has some very interesting applications to how we're going to find therapies and treat individuals successfully. And we can have a little discussion about some of those things. So I always consider there are certain things that make us vulnerable, and there are certain things that make us stronger. So we'll talk a bit about the cellular pathways of vulnerability. You may already have heard about excitotoxicity, and where too much glutamate is being secreted on the motor neurons, and that is the target of the drug riluzole. What I'm going to focus on mostly today are misbehaving proteins and how the cells manage them and how we might manage them, as well as transportation issues throughout the cell, loss of the connections and regeneration to other cells in the network, including brain and muscle. And then review some of the cellular defense mechanisms that cells have to defend themselves against these vulnerabilities, and also the influence of aging. And as I mentioned, strategies for mitigation. So just as a little review, this is on the left is the nervous system from the back. So the brain, the cerebellum, the spinal cord going down, with areas where the motor neurons are that go to the legs, to the arms, back to the arms, and then up in here to the bulbar region, the face and the eyes. And so these motor neurons, this is a depiction of one, there are collections of them in these nodes. They have two kinds of processes. They have dendrites, which project out into the spinal cord and interact with all kinds of other different neurons, receiving their input and instructions. And then the output, which is the axon, which goes, in this case of the lower motor neuron, to form synapses with muscle. And in case of upper motor neurons, which receive the information from the frontal cortex and elsewhere in the brain to design the movement, they then synapse down directly to the lower motor neurons or through different relays. So what happens if we look at a healthy, happy motor neuron, this would be a section through the spinal cord here, like that, and stained so that we can actually see the neuron. There's its nucleus, here's its shape, these are the dendrites going out, and here's probably the axon here. They're big, and they have these extensive dendritic trees, and full of these cytoskeletal elements called neurofilaments that, form tracks and keep it strong and sturdy. This is a motor neuron on ALS. Look at these dendrites, they're all shrunken, and this axon has a big balloon in it where some neurofilaments have accumulated. These pictures were from Sterling Carpenter, who was the neuropathologist at the Neuro when I went there, and he managed to get this section that showed so many different features of an ALS motor neuron, but they wouldn't all look exactly like that. This shows you some features. So they get sick and dysfunctional, they shrink, the dendrites retract. I kind of think of it as, when you've got the flu, you go to bed and you put the covers over your head and you say, leave me alone. That's kind of what the neuron does. And the neuromuscular junction gets denervated, keeps trying to reinnervate, but eventually these things fail and the motor neuron becomes dysfunctional and then dies. And a major question while I'm on this topic is that even though the motor neuron may be dysfunctional, it might not be dead yet. And I think that is just because of it. If any break in this conductivity means the muscle doesn't get the message. So it might be possible that there are neurons that can be saved if we could find the right treatment. Before there'll be some that are on their way out, but yet. Okay, so in terms of variability, it's tremendous. So here's a graph, a number of people in a particular study, their age when they first were clinically diagnosed with ALS, and it goes from 20 years old up to almost up to 90. With this distribution in the middle, which is why we say that the average mean onset is 50 some years old. But, these are, it's a tremendous variability. And if we look at the variation in the progression of the disease, how long after clinical diagnosis does it go on? Again, a huge variability. This is the ALSFRS score, and it goes anywhere from people who have succumbed quite quickly to this guy over here who, lived for a very long time and then crashed. Of course, we don't know why this person crashed. It could have been 84 years old, could have been whatever. But just that variability also look at some of these where there's like a hump that looks like things are getting better, and then they come back down, and then whoops, doing that. So it's kind of a bumpy ride. And the difficulty is that, if this person were on a drug, you wouldn't know if this was the natural history of that person's disease or if the drug was working. So you have to kind of keep going and see. There's also the variability in the pattern of the motor neuron involvement. So some people have first symptoms in the legs, others in the bulbar regions, a mixed, and that also is going to affect not only probably some of those slopes of those lines in the previous ones, but also therapeutically, if the person's disease isn't at the same stage, even in the one body. So if you start with a problem in the legs, those neurons are going through those different stages of disease before the ones, in this case, in the bulbar region. People are still speaking and swallowing and having no problem there. So do we need the same drugs for these different parts of the body? Or do we need combinations of things that will try to do something with the lower motor neurons, in this case, and preserve those that are in the brainstem or in the thoracic region? So these are just some of the considerations that we have to deal with when we're trying to figure out how to do therapeutic trials. So even when we know the single cause of the disease, say it's a mutation in SOD1 ALS, this graph shows the age of onset. This shows different mutations in SOD1. But even if we look at one mutation, all these dots represent a person. And look at the spread, 25 to 75 or 80. Same mutation, and even within the same family, one can see that variability. Same with the duration of the disease, quite variable. So where we are here is that the clinical manifestation of disease is influenced not only by what causes it, but by the response that the cells and the organism have to it. And that some factors will increase the vulnerability and speed up the disease, and other factors are defending and compensating. And the effect of such causative factors is widespread, involves many multiple cellular functions and interactions, and we'll talk about a couple of them. The inter-individual variability in these factors, and the intra-individual variation in the disease state within the axis, which I just referred to, and how that poses difficulties in testing and applying therapies. Because if the success of phase 3 clinical trials has been rather abysmal. We have things that we try in models, whether they're cell culture or animal models or whatever, and some of them even look like they're promising in initial phase 2 trials. And then we do a big phase 3 trial and nothing works. But apparently nothing works. So we might be missing things, because with all that variability that I just showed you, could have some of those people who are actually benefited, but they just it's overwhelmed by the data from the people who don't. And we're starting to look more into those kind of issues and looking at phase 3 trial results, post hoc as we call it, after the fact, and try to find out if there are people who actually did seem to benefit and what their characteristics are. So this is kind of what I've been doing for my career, is looking at these factors that are stresses on motor neurons and how that we might, support them with all these defensive mechanisms, like the body's been and cells have been doing all along, but just, prop them up and, keep going. And before it hits that kind of precipice where I say everything just goes to hell, and the disease progresses very quickly. So again, we have the glutamate receptors and excitotoxicity. We actually way back showed in a culture model that if we block glutamate receptors on motor neurons, they didn't get inclusions of SOD1 and they lived. But of course you can't do that, because if we block the inputs to your motor neurons, then you can't move anyway. So, but it's just kind of a show of how that stress actually contributes to this living on the edge of motor neurons. What I'm going to concentrate, and in terms of support cells, there are the glia that are surrounding them, the astrocytes. They're giving them antioxidants and factors. So the motor neuron is kind of like a princess that sits there, and these cells around give them goods like glutathione and these other antioxidants and support them. And microglia are part of the immune system that have factors that support them, but then they turn ugly and try to take them out. So that's a whole webinar in itself, and maybe we can get somebody to talk about that aspect. What I'm going to focus on today are proteins. Proteins in the cell that form inclusions, and these chaperones that protect proteins, and proteolytic systems that degrade them. And because that'll be enough. Now, I'm going to try to, this is not for you to read, it's to show you that almost everything in the cell, here's our neuron, here's our nucleus, here are those glia that I talked about, there's the excitotoxicity, but there are so many processes in these cells and functions, and there are mutations in genes and encode proteins that are involved in all these different cellular mechanisms. So it just shows you that so many things can go wrong, but yet we have the same result, that we have a motor neuron disease. And sometimes we'll have a cognitive disorder that goes with it. And so if we can find some of these commonalities and devise interventions for them, such as the proteins I'm going to talk about, and mix and match therapies that would tackle the different problems like mitochondria and transport in the cell, etc., then maybe we'll be able to make some better headway in this way. So proteins, I'm going to try to be very simple in this biology, but I told Donald that if you have questions where you don't understand something, to bug him to interrupt me, and we can explain it. This is kind of a little proteins 101, hopefully. But what neuropathologists have seen for a very, very long time, since we started looking at tissue from individuals who have these neurodegenerative disorders, are these big gobs of proteins that are in the cell. So in Parkinson's, we call them Lewy bodies. In Alzheimer's, there are amyloid plaques and neurofibrillary tangles. In ALS, we have these ubiquitinated inclusions, all kinds of different ones. And even in the familial forms of ALS that you may be familiar with, like SOD1 mutations, we have big gobs of SOD1 sitting there where it shouldn't be, that is aggregated. The same with some of the RNA binding proteins like FUS. And in familial ALS due to TDP43 mutations, we see them, but we also see them in sporadic ALS and some of these other forms where this particular protein is very amenable to not being where it's supposed to be in the nucleus and getting stuck in the cytoplasm and forming these inclusions. So just a little reminder about DNA to RNA to protein. So this is one of my cultured motor neurons. Kind of looks like the one I just showed you from the spinal cord. Here's this nucleus. Here's a portrayal of the nucleus. The DNA is in the nucleus, encoding all the genes. And then that gets transcribed to pre-messenger RNA, which is, a copy of the template that can be used to make protein. And in the nucleus, there are some changes that happen to fine-tune that RNA, including splicing out little bits that aren't supposed to be translated into the protein. It comes out of the nucleus and goes to this thing called the ribosome, where the ribosome kind of reads the code and sticks in the appropriate amino acid for that codon on until it starts making this chain of amino acids that is going to be the full-fledged protein. So now proteins are unique in that each protein is unique because it not only has a unique sequence, but it will take on a unique shape according to that sequence. That shape has to be formed for the protein to work, kind of like a lock going into a key. It has to fit. So The first, what we call the primary structure, is that amino acid sequence. And then within that sequence, there are regions of amino acids that have particular interactive protein or properties with other elements of the protein. So it kind of will fold on itself, and those will interact and stabilize. And then the protein will take on its kind of normal full structure and have its final shape. And some proteins will form complexes with other proteins. In this case, we have a dimer, two of the same guys, and SOD1 does that. It forms dimers. And then we have other protein assemblies that are very complex, of many, many different proteins that have to be there at the same time, have to interact appropriately, and form these organelles that are function. So there are many things that can go wrong through this process. And if it doesn't go right, then we can get these proteins aggregating and sticking to things they shouldn't stick to, gumming up the works, and eventually forming these inclusions. And so the causes of, say, these abnormalities that I showed you in the last slide is, first of all, the sequence could be wrong. So you have a mutation that increases the protein's propensity to aggregate. Now, all proteins will aggregate. If you give them concentrated enough and leave them long there enough, or put them in the right conditions. But some sequences are way more prone to aggregate than others. And interestingly, those proteins tend to be the ones that are mutated or form these inclusions in neurodegenerative disease, because there's already a propensity there. They're vulnerable if all this control system doesn't go right. Environmental stresses. So oxidative stress can oxidize some of these guys and then cause them to behave improperly, and the cell to perceive them as being not normal. Also, when that process I just showed you, the cell's making mistakes all the time. All that transcription translation doesn't always go like it's supposed to. And there are mechanisms that we're going to talk about in the cell that detect that and take them out and leave the good and focus on the good ones. But if and they have to, every protein has its own cycle. It only lasts for so long in the cell, and it's time to make new ones. So if that handling of the quality control of the proteome of these proteins is impaired, then we can get all this aggregation process happening too. So here's our ribosome again. Here's our little protein being made. Now, I said there are these protective guys. Well, the first ones are called chaperones, because what they do is they're these kind of barrel-shaped proteins and they kind of hug the chain as it comes off and protect it from all the other things going on in the cells, where it doesn't stick to where it's not supposed to stick. Proteins don't come in and interact that aren't supposed to until it gets formed, kind of like chaperoning your kids until they're grown up. So when that comes off the ribosome, then the chaperones let it go. It folds nicely, but it doesn't necessarily stay that way. It's kind of like the forces will fight back and forth. So the chaperones can come in and refold it as well. But if that doesn't work, then there's a process to take it out. And in the case of a mutation, it's going to be harder for this process to work. Not impossible, but harder. And if the oxidative stress, the same issue. But if it doesn't get taken out, then it's going to aggregate. But if it's supposed to be taken out, then it gets this protein called ubiquitin stuck to it. And then what happens is it gets taken to these organelles that dispose of it. So there's where we were before. So the proteasome is kind of that I showed you a picture of that when I was talking about proteins from the side. But it's basically a barrel, and it feeds the protein down in, and then chop, and spits out pieces at the bottom that can then be reused to make new proteins, or they might get spit out, or, whatever. There's like a disposal recycling of the machinery of the cell. Now, the aggregates can be a bit too big to go in there, so there's another process that targets them by a process called autophagy, auto eating up, and fusing them with these organelles called lysosomes that have these acidic enzymes in that just do the same thing, cut the aggregate proteins into pieces and dispose of them. These things can also get secreted and actually then even make their way into the bloodstream, where we can, use them for biomonitoring to see what's in the blood. Are there abnormal proteins in the blood? And the chaperones also get secreted because they have to protect this space out here too. So I hope that gives you a kind of an idea of how, there are chaperones that protect these proteins, and then there are these mechanisms that get rid of them, either because they're just old or they're damaged, or because they're now becoming toxic and we've got to get them out of there. So what happens at ALS? Well, the chaperones and proteasomes get overwhelmed because this amount of these abnormal proteins, the amount goes up, and they keep trying to misfold, and they have to keep refolding them. And then eventually they aggregate, so they got to get rid of them. So it puts a stress on the cell. And the other thing that happens is that the proteasome levels actually decrease rather than increase to take over the load. And these misshapen proteins accumulate, misbehave, act inappropriately. They bind to mitochondria, they bind to the surface, they just bad characters, and aggregate. The aggregate itself might not be toxic, but it starts going up the works. So this, I'm going to show you an example. This is a lot of that work that led into that previous slide was done in my lab, but also others. And here I'm also going to apologize to the other scientists, because I'm not referring to everybody's, all the people who contributed to all this knowledge that I'm trying to portray to you. So again, if we're looking at a section of the spinal cord, see the dark guys, these are motor neurons, and they've been labeled by a component of that proteasome. And you can see that dark means there's a lot of it. And you can see how dark they are in the control. Here are some more. But then we look at ALS spinal cord, and they're much fainter. In fact, sometimes you can hardly see them on the background. So that's what I mean by the levels of the proteasome go down when they should be going up to get to protect the cell. And not only does the whole proteasome not increase. You got a question or something? Are we okay?
Yes. Sorry, Heather. I didn't.
That's okay. That's okay.
Trying to process all of this in my head as well, and also trying to keep track of questions.
Anyway, I just wanted to. It's not intuitive.
Yeah, I know. Sorry,
So
we had a.
I'm losing you. Well, hopefully I don't lose you too badly.
Could you kindly expand on what is meant by environmental stress? That was a question that came up about 5 minutes ago. My apologies.
Okay, no, that's right. Could we keep that one till the end? Just
Yes.
make sure that's the first one coming up, because then something we might discuss, rather than just, could be anything like toxic proteins, like insecticides or something, so that's the concept. But keep the questions coming. So not only would have less proteasome, but I think I mentioned that this proteasome is now composed of all these different subunits of different proteins, and they gotta be there at the same time to get together and properly and be efficient. But what we find is some of these proteins aren't there as much as they should be. So one of them, beta 5, another one, 19S, isn't as much of it in the spinal cord as the other subunits. Whereas in the cerebellum, which is far less affected in ALS, everybody's there cooperating, making proteasome, and everything's fine. So then we get to, okay, so, we're particularly with people who we know have a mutation that is very penetrant to give ALS, they're not showing symptoms of 25 or even, 50, 75 years of age. So there's, they're protecting themselves. But as we age, our ability to do those protections is compromised. We can still manage, hasn't reached a tipping point, but our cells are not as efficient. The first time I saw from Sterling Carpenter a section in the spinal cord from a 50-year-old and saw the gobs of things in the motor neurons of apparently normal person, didn't die of neurodegeneration, thought, oh my God, that's how my neurons look. Because some of these things happen to a certain extent with aging because of mistakes or whatever, but it's not enough to really cause us too much trouble. But we also can activate those stress responses. But that ability to do so diminishes with aging. All those things I just showed you are also natural aging phenomena. And it's interesting that people who can upregulate chaperones and keep them where they need to be and have good stress responses tend to live longer. I just referred to that about the complexes needing to be coordinated. And about a third of our proteins become what we call metastable. They have some issues with aging. And that's that decoupling of their production with their other partners in assemblies that I referred to with the proteasome. And again, that the proteins that are most prone to aggregation, that actually the cell has to fight to keep them from aggregating, are the ones that are found in ALS. And even this thing called the bystander effect is very interesting, because what it says is that we all have these polymorphisms and different sequence. There's all a sequence variability in our genes as an individual, and some of them are no problem. Some of them actually make the protein work better, and others maybe compromise that function a certain amount, but not enough to really cause us a problem. But if we have a lot of those kind of vulnerability factors that we can call them, they actually influence the aggregation and toxicity of those major disease proteins. So this is where we need a lot of work, is understanding what aspects of our genome are actually making us vulnerable, and those that are actually making us protect us from those aging and disease factors. There are also other things that happen, which I don't have time to go into, but there are certain regulations on DNA and RNA that influence whether they're transcribed or not, or work or not. But that can also be an influence, but I won't take you there. You'll hate me. And organisms that retain a robust stress response live longer and better. I can remember a study I read decades ago that actually got me interested in looking at these stress proteins. And they had taken lymphocytes from the blood of people who lived over 100, centenarians, and compared them to other age groups. And they took these lymphocytes and they heat stressed them. And what heat stress does is it increases the production of those chaperones, and that's why they're called heat shock proteins. And that ability to induce those heat shock proteins, according to the stress, is one of those major factors that keeps things going. And those people who were over 100 retained a very strong stress response compared to those that didn't. So that's interesting. It also leads us to talk about what strategies can we use to improve those defense mechanisms, like increasing the chaperone activity, making them stress responsive, or helping the proteasome and the lysosomes, etc., get rid of them, or reduce their production. So reducing their production I'll look at first, because that's what we're doing with antisense strategies with familial ALS. So individuals with SOD1 mutations, there's the antisense oligonucleotide that interacts with the RNA so that cells don't make as much SOD1, because they've been coping all those years with the amount they had, and then they end up not being able to cope. So if we can reduce that level, then maybe the cell can go back to above that tipping point and cope, as long as there's enough to do the normal function of the protein itself, which is an antioxidant. And it turns out that with SOD1 ALS, one can inhibit that production quite a bit without seemingly cause a big problem to the function of the organism. So this is a real success story. Yeah, we have someone on our board who is doing very well on this. And maybe there are some people listening who are involved in, are getting that therapy. Some of the others, like FUS, which is an RNA binding protein, those have to be retained in very strict levels, not FUS and TDP43. So the strategy can work, but it's a little trickier because we can't go too little either. We can't go too much. But the other thing that we've been trying to do for years and still haven't managed is to get the cell to make more of those heat shock proteins. And you may remember that there was a drug called arimoclomol from a few years ago. And what arimoclomol does is it interacts with the transcription factor on the gene for HSPs, heat shock proteins. And when stress happens, they keep that transcription factor on longer on the gene, so it makes more RNA and the idea is it makes more heat shock proteins. But they're not made all the time, they're made as needed, which means it's not going to be toxic. And so arimoclomol is supposed to do that. And it showed some benefit in a mouse model, and a little bit of an effect in some culture work that we did, but it didn't work in a phase 3 trial. And I think there are a couple of reasons for that. And one is it's not a very good inducer of heat shock proteins. We recently published some studies a couple of years ago where we didn't see any increase in heat shock proteins in mice in either the SOD1 or FUS models of ALS. It does some other things that are interesting, but it really is not very good. The other thing that I'll get to is we need to be able to know that it actually is doing this job when we give it to the person. We need to have a marker of whether those heat shock proteins are inducing, like we did in our animal studies. There are other drugs that turn this system on all the time. And so, yes, they make more HSPs. They've been shown to do that. But the problem is it doesn't last. They desensitize. And when they don't, there's toxicity. These are cancer drugs. They're used to, they have other effects that kill cancer cells. So trying to use those is a bit tricky. Another interesting idea is what we call chemical chaperones, ways to actually target specific proteins. An example is our antibodies that have been raised against TDP43, which is one of those proteins that's mucked up in all kinds of ALS and forms inclusions. So the idea being this antibody is designed to only interact with TDP43, and then it binds there, and then it won't be able to cause problems. There are also small molecules being invented that target TDP43 and other like molecules. And there's one called JMRS from Janice Robertson at University of Toronto, which is kind of interesting because it seems to have an effect on a lot of different things, not just TDP43. So we'll see where some of these go. But there's also this issue of, okay, everybody sees TDP43 aggregating, in ALS. But when we do experiments, we see what we look for. So if we only look for TDP43, we're only going to see TDP43. But what about other things? What's the totality of what's going on? And will targeting TDP43 be enough? And I guess that's what we'll find out. So basically, that's that for proteins. But these things also have to get where they need to go. And that's at the second short part that I'm going to talk about is the transport from the nucleus to the cytoplasm is messed up, transport into the axon is messed up, transport into the dendrites is messed up. And if we can't get constituents that are made in the cell body or even the constituents to make them distally into these processes, then we're not going to have a good functional neuron. And it's going to do that, go falling back. So if we think about how we might, and when we were looking at the role of chaperones in stress, we started to realize that the motor neurons had lots of stress proteins, they had lots of chaperones, but under stress, they weren't necessarily getting up where they needed to go. So they weren't getting up into these synaptic regions to protect the synapses. So it's not just a matter of how much, it's where you get where you need to go. And the inclusions can also, form impairment in this transport systems. Now there are one of the things that happens with TDP43 being trapped out here is that there's that process of MRI processing I referred to, where bits are taken out that aren't supposed to be in the final protein. That doesn't happen. They call them cryptic exons that get contained still in the final protein. And so there are studies going on to look at how manipulating TDP-43 through Stathman, these proteins called Stathman2 and UNC13A that have these cryptic exons left in them could be kind of restarting the transport systems and making them work properly, because Stathman2 is important for transport. Another thing is not just the gene, is whether the mRNA is whether that protein is getting made. And I showed you an example of the proteasome that isn't getting made well, and that there's kind of this general impairment of that translation process. There's also the issue of connectivity, because as I mentioned before, if you don't keep, there's the motor neuron axons coming out. There's one motor neuron and it's putting synapses in all these muscle fibers. There's another one in different muscle fibers. And if, say, this motor neuron gets sick and retracts its synapses, it'll try to regrow them, and it will succeed a lot of times. Oops, okay, try again. Okay, contact, back, up, contact again. But eventually, these guys are left to their own device. These are left alone, and they're not getting the instructions to move. But this neuron is nearby, and it'll take over. It'll send processes out, and they'll have all these will now be green. But the problem with that is, well, the good thing is that those muscles will still function. The bad news is now this green motor neuron has way more work to do to manage all of these guys as well as what it was supposed to. So it puts more stress on the system. So the defense is, innervating, denervated guys and reinnervating the muscle fibers. The workload gets more, and the more stress and eventual collapse. So there are strategies being tested to try to keep these synapses connected. And Richard Robitaille at the University of Montreal has a molecule that he's testing for that. So this is the summary. Basically, if you got enough of that, what I just said, that biology, you would hopefully have understood that if you make this folded proteins that aggregate, you got to get rid of them. And that this depends on a coordinated effort of multiple chaperoning and proteolytic pathways. That these quality control systems are inducible to match levels of stress to keep things going smoothly. Some cells, some people do it better than others. If you have a defect in the protein folding or proteolytic system or a poor inducers, you're invulnerable, like those other polymorphisms that I talked about that normally aren't a problem. Same goes for oxidative stress, antioxidants, and all those other things on the list of that slide like this I showed you before. The bad news is it doesn't get better with age, it gets worse. Oh, whoa. But longevity is associated with maintenance of good stress responses. So small stresses actually activate these systems and protect you against bigger stresses that might kill you. And this process is called hormesis. So you can think that some stress is actually good for you. And what we need to have more also work on is what kinds of work we can do here so that, understand if there are certain stresses that actually can activate some of these protective mechanisms a little bit, but then the cell kind of wakes up and says, okay, get busy and protect against everything. So that's an interest of mine. So these are some of the people over the years that have contributed to the work in my lab. I'll show you a couple of few examples. The funding that we've had, one of the greatest things was the Hudson Translational Team Grant from ALS Canada, where we worked with Josephine Noglantoglou and Chantal Sefton and later on Maria Vera to look at some of the things I talked about. And of course, Muscular Dystrophy Association, ALSA in the US, eRare. Canada is considered part of Europe for the rare disorders granting. Seems we're following up on that politically as well, that we like to have associations with Europe these days. And thank you for your attention. I leave you with why I live in BC.
That's fantastic. Thank you so much, Dr. Durham. That was very informational. Is that the right word to use? It was educational, informational. It was very eye-opening. Again, we're so lucky to have researchers such as yourself present on stuff that you don't normally find, or sometimes if you Google things, you never know what you find on the internet, right? So to have that presentation is very meaningful. There was two questions, well, maybe three questions now. The first one on environmental stress. Can you kind of expand on what is meant by environmental stress?
Mm-hmm. So maybe I could start by, I think it refers to things in the environment that we are exposed to that might put those stresses on our body and might be particularly damaging to the nervous system. And we often think of things like pesticides. We see that there are clusters of ALS in certain occupations, such as farmers or soccer players or football players or people who've been to war or whatever. So trying to understand what factors might be predisposing to tip those systems over the edge or put that stress on is we really need to know that. But we have a problem in that this is a rare disorder. And if we look at the numbers of people that were necessary to show that smoking caused cancer or someone who's got heart disease, it takes to do these kind of epidemiological studies where you try to make associations between somebody with a disease and what they've done with their life. It's very tricky because you make correlations, but correlation isn't causation. So we know that farmers can be exposed to a lot of pesticides, spraying the trees, etc. But it doesn't mean that that's what's contributed to their ALS, or it could. Sometimes we see clusters like there's one around. Ellen was talking about people around here in Victoria that they're a cluster around particular farms. So we have to investigate these, but how do we know what to do? Athletes, interesting. There seems to be a lot of athletes, and that's because we just hear about them because we follow them, or is there some predisposition? Is there something about your motor neurons that they're very active, and that makes you a good athlete, but also puts a stress on you? Yeah, all these kind of questions. But how do we answer them accurately when we don't have big populations to make the correlations, because correlation isn't causation? Does that help? Well, we know certain things like metals. I think that's probably when I started as a neurotoxicologist, and we used to study neuropathies that happened when people were exposed to large amounts of pesticide or drank adulterated liquor or whatever, or worked in shoe factories where there was glue smell was everywhere. And there it was very easy to make the correlation of their exposure to a particular disease because it was so obvious. And there were so many people who did that, but then the people outside didn't get it. But now we have occupational health and safety. And so we don't have as much of this in Canada or in North America, at least. We're not sending people down into the coal mine unprotected. So we still don't know if there are exposures that might be influencing disease. And what disease it causes may be due to the other vulnerability factors. So you can see when you really start looking at what a mess it is of how to sort this out. So just try to take care of yourself and get exercise, eat properly, don't expose yourself to toxic stuff. And if you live by the ocean, look out in the evening and feel calm.
Another question, why do patients receiving tofersen also take riluzole? Is there any benefits of taking both?
I would say so. First of all, we know that riluzole can help a little. It's not magic, but it can help a little. And we also know that it takes tofersen time to work. And they're really seeing, what's really interesting is that they're really seeing the benefit after a fair amount of time. Because if you think about that slide I showed you with all those different the mess of everything that's happening, that's going on in there. And there's also the immune system that's been attacking these cells. So even when you reduce the amount of SOD1, it's going to take time for all that to calm down and start working properly. So it kind of makes sense. But in the meantime, you don't want to stop taking something that might be helping that bit of excitotoxicity. And it's not it's a very safe drug. And when something's working, you really don't want to tell somebody to stop taking it, when it might be helping them. So that would be my answer.
Great. One last question. This patient has been 4.5 years since their PLS diagnosis. 10 months ago, he sustained a relatively mild pontine stroke in the fine blood vessels of the pons due to very high cholesterol. Prior to this, my speech was completely unaffected, but now it is a bit, plus my balance is worse. Have I essentially been pushed further down the timeline of my PLS?
Well, looks like there's more than one disease. So how those would interact is a good question. I mean, if there's hypercholesterolemia and there's been a stroke, all that may have happened anyway, but it's also putting a stress on the body, and if it's affecting some of the systems that are with ALS. So I'm not a physician, so I can only kind of conceptualize. I'm not going to give advice or say what it is, but I guess that time will tell this person's probably being followed by a clinic, and they'll be repeating the functional scores, but it'd be something to look for as to whether the stroke system stabilize and improve, or things get worse. So unfortunately, no way to predict from that, but there's no reason also to say all of a sudden everything's going to hell.
Right. Yeah, very, very true. Another question, is there a test to measure the effectiveness of tofersen?
It's based the effect of tofersen at the level of SOD1 protein, or the effect on the person. That would be what I'm wondering. That's what the question is.
Yeah, maybe the person. I mean, having been
if it's the person, that person, if they're on tofersen, they're being followed by a neurologist, by a multidisciplinary clinic, and they'll keep doing the same measurements that they were doing before, and they'll see whether those stabilize, or even after time, even improve a little. So the proof is in the behavior, basically.
Amazing. What is tofersen? Has never been prescribed for my wife. Sorry, let me I think the question is, what is tofersen? And that's a drug specific to a type of ALS, genetic more than anything.
Yeah. So it's specifically for patients who have a mutation in the gene for the protein that encodes the protein SOD1. And that I showed you an example of that in one of the slides. But so the idea being that if the cells have a lot of SOD1. It's a very highly expressed protein and a very stable protein. So the cells have managed for a long time with that level. But if you knock it down by preventing so much from being synthesized from the RNA, then the cells can try to cope with that a lot better. So what an antisense oligonucleotide is kind of like a complementary sequence to the RNA. So it's like something that sticks on the RNA, which is needed to make the protein, so it can't make the protein. So it's a blocker of the RNA to the protein, if that makes sense.
Yeah, and the reason why some patients may not be prescribed tofersen is because it's specific to one type of ALS, and so if you haven't been prescribed it is not proven to be effective unless it's SOD1 ALS.
Yeah, so if the person knows whether or not they have SOD1 ALS, if they have it, then that's a discussion with their clinic neurologist. If they don't, then it would be totally inappropriate. Correct. If they don't know, then they should be associated with an ALS clinic that could provide them with genetic counseling. And we have ALS Canada has recently funded a nationwide genetic counselor that clinics can use, but clinics also have their own people that they use in their center.
Great. Another question, what are your thoughts on fasting?
I will be hungry. No, so I don't mean to be flippant, but fasting, I don't know. I think we don't know enough about, I don't know. The whole issues of diet, exercise, we really need more people looking into that and need somebody to come on and talk about what is known. There's some interest now in the ketogenic diet, fasting. I guess it depends how much fasting, and I don't know if we can find that out. If it's, a short period, I mean, they say we should fast overnight for at least more than 12 hours or whatever. But if this person is saying, well, I'm going to fast for 2 or 3 days or a week, I don't know. The other thing is that ALS patients eat a lot of calories, and that would be my concern as well. So if it were me, I'd eat for that reason alone. Their metabolism is more revved up in people with ALS, and people tend to be thin. You need to keep those calories up, and even when you can't, it starts to become difficult to take in food by mouth and swallow. It's start feeding tubes early. So even when you're still enjoying your food, so you can enjoy it, you can get that nutrition because you need it. Your cells need it. Your mitochondria need it to fire up your cells. Mm-hmm.
And certainly, please consult the dietitian for any further advice on diets and that.
Yes, thank you for saying that. Yeah, I should have said thank you.
Right. Dr. Durham, we're at the top of the hour. Thank you so much, and we're out of questions as well. So just wanted to take this opportunity again to thank you so much for the information and sharing your knowledge of your decades of knowledge that the research that you've done. Congratulations on all of that. I don't think it's stated how world-class the information that you delivered and all of the research that you've done through your career. It's just so well respected around the world. And we're very lucky to have you in our backyard here in Victoria. So thank you for sharing your knowledge and spending your afternoon with us today.
Well, thank you very much. And it's a pleasure. And I can't quite quit yet because I still haven't fixed anything. So we keep thinking we can fix it, and then we haven't. So I'm not giving up yet. But also, if anybody has further questions or that it's a lot of biology I threw at you. If there are questions, I'm happy to answer them. So you feel free to refer people to me.
Yeah, no, and we can certainly connect you with Dr. Durham. Please connect with Alexandra Guerrero, who has been helping out with obviously the webinar as well. Our next webinar will take place on Tuesday, February 10th. A presentation from Daniel Goldgut, the co-founder of Epilogue, who's going to talk on the important information on writing a will. So please make sure you do join us for that. And I'll reiterate what I mentioned at the start. These webinars are for you. Let us know what speakers or topics that you would like to hear at future webinars. We do have 12 planned 11 more, I guess, for 2026. And certainly we want to find topics that are interesting to our viewers. So please don't hesitate to contact Alexandra. If you do have any suggestions on topics or speakers, we're happy to look into those. So on that note, thank you again for joining us today, and we look forward to seeing you next month. Have a great afternoon.
Bye, everyone. Have a good afternoon.